Digestive system. Digestive system is what we use to eat. Why do we eat? Well, in the day-to-day um, processes inside of our bodies, we lose energy. We dissipate it in various ways, like as heat or as part of molecules that we are getting rid of in various ways through sweat and saliva and feces and urine. Uh, those The energy bound in those uh, chemical bonds of those molecules has to be also replenished. Uh, those are the molecules that are metabolites of various biochemical processes in our bodies, and those metabolites are kind of molecules that we cannot reuse or transform into something we don't need. So we need to take energy in various forms, including in the forms of various molecules that we need to replenish our own stores, our energy stores, both in the form of heat and in the form of chemical bonds of different types of molecules, and we need different types of molecules in order to replenish molecules of our own that we need for our day-to-day cellular processes. Uh, so, as you remember from the very beginning of the course, one of the main definitions of life is that the living thing can take in, store, transform, use, and dissipate energy. And you also know that most cellular processes require energy, and that energy is used. Those biochemical processes can use the energy if it's stored in phosphate bonds of ATP. And cells make ATP by taking energy out of chemical bonds of glucose, and glucose has to be obtained from the outside world. And so that is the main job of the digestive system. Now we don't have to take in pure glucose. We can take in other types of carbohydrates, monosaccharides, disaccharides, or polysaccharides, and break them down and transform them into a monosaccharide like glucose that we can break down in our mitochondria. We also take in from food other types of molecules that we need to replenish our own, like lipids to make our own, let's say, lipid membranes or steroid hormones. We need to take in proteins that we then break down into amino acids because we need those 20 essential amino acids in order to make our own proteins. And proteins are going to do all sorts of jobs in our bodies, enzymes and structural proteins and various other roles. We need to take in nucleic acids in order to get the A's, T's, C's, and G's. Much easier to take them in than to build them ourselves in order to build our own DNA and our own RNA. So, what we are taking in are foreign molecules, molecules that come from plants and fungi and animals or synthetic foods. Whatever we are eating is going to have those types of molecules that then the digestive system is going to break down into the essential building blocks, the smallest building blocks. So we could build our own molecules, our human molecules, out of those building blocks. We also need some minerals in order to uh, introduce them our into some of our molecules. Some of our molecules need iron, like hemoglobin. Some need selenium or sulfur or phosphorus or calcium. So we need to take some of those minerals out of the food because some of those are going to be lost in various secretions coming out of our bodies. And finally, there are some molecules specifically that we cannot make. Those are called vitamins that are essential to aid in some, as enzymes or as molecules that are aiding in particular biochemical processes that are essential in our body, but we cannot have them done without the presence of those vitamins. Uh, so, what is important from what I just said is that the proteins, lipids, carbohydrates, nucleic acids, and all those molecules that we're bringing in through food are coming from foreign materials. They're organic materials, but they're foreign. They're not humans. They're not exact shapes and sequences as those of our own body. So digestive system has the job to break them down into the simplest building blocks, and then our body can use those simple building blocks to build our own proteins and our own carbohydrates and nucleic acids and lipids. It is like eating a cow Lego truck, and digestive system is going to break it down into the smallest Lego pieces, take it into the bloodstream and liver and muscles, and then our cells are going to build human Lego trucks out of those little Lego blocks. So it really does not matter what shape or what sequence those are coming in from. They can be completely synthetic. They can come from plants, fungi, animals, protists, whatever you want to eat. And depending on what you have, what you like, where on the planet you live, what is the season. You can eat all sorts of different things. You can also eat things that are mixes, like various GMOs. It doesn't matter because your digestive system is going to break it down into the smallest, most essential building blocks. And it really doesn't matter what is the sequence of that protein or DNA or RNA of the food you're eating, because none of it is going to get into your cells before being broken down first. The digestive system consists of the GI tract or alimentary canal and accessory organs. Both of those are equally important for proper digestion and for meals and thus for survival. Alimentary canal is a long tube that starts with the mouth and ends with the anus. The food going through the canal is never passing through any cell membranes, right? It goes in the mouth and ends up in the anus, and as it is going through the body, it's always outside of our cells. The food is always outside of our body. So the lumen, and whatever is inside of the GI tract, is outside of our body. It's officially, technically, the outside world that is just making a pass through us. Uh, so it is the cells lining the alimentary canal that are providing a barrier between the outside world that is passing through us and the inside world, our cells. So there are different types of epithelial lining the GI tract all throughout. And your textbook is pretty good, and the lab manual is pretty good. It's telling you what kind of epithelia are in different parts of the alimentary canal, what kind of cells you can find there, including various secretory cells, immune cells in different parts of the GI tract. Uh, so the food that we eat might also contain, toxins or things that we cannot digest. It may contain too much of something. It may contain just dirt and dust, uh, and those kinds of things cannot pass, or usually cannot pass, through the membranes of the epithelia of the alimentary system. Those things do not have evolved transporters that are going to transport them across the membranes. And so they're going to remain in the lumen of the alimentary canal, and they're going to go out into the outside world through the anus as feces. Uh, so most of the stuff that we don't need, don't want, can't process, that is toxic, that is dirty, is going to just pass through us and never get absorbed by the intestinal wall cells. Uh, the GI tract has several segments, and each segment is tasked with processing food in a particular manner. So first we have the mouth, then the pharynx, then the esophagus, the stomach, a couple, three different parts of the small intestine, and a couple of different parts of the large intestine. The accessory organs are the teeth and tongue and salivary glands. Those are all in the mouth, the liver, gallbladder, and pancreas. Each one of those accessory organs has a role in processing food in one way or another. Um, so, digestion involves several processes. Ingestion is taking in the food. Propulsion means moving the food down the canal. Mechanical digestion means that there's going to be some way of taking tough big pieces of food and breaking it down, turning it all into liquid. And then chemical digestion, which means enzymatic, the breakdown of the large molecules in the food, making turning it into the smallest possible units that can then be involved in absorption. So cells of the intestine are going to absorb those tiniest molecules, take them in, put them in the bloodstream, and into the rest of the body. And the final one is defecation, getting rid of everything that was not digested and absorbed. So, yeah, ingestion is eating, the way the food enters the alimentary canal. Propulsion is a set of mechanisms that moves the food down the alimentary canal. The proper name for that movement is peristalsis. Mechanical digestion. Teeth are breaking, cutting it down, and the tongue and the teeth together are turning the food into a soft, but still kind of semi-solid bolus. And then once it passes the digestion in the stomach, which is also churning and doing mechanical digestion, it's going to be turned into liquid called chyme, and that liquid is then going to move by peristalsis through the rest of the intestinal canal until it goes out. Okay. Um, so um, digestion requires the food particles, all the food, to be suspended in liquid. In order, that's the only way the enzymes can access the molecules and break them down. Uh, this is why the alimentary system, all of it, from the beginning to the end, including the accessory organs, puts in a lot of water into the GI tract. Water mixed with all sorts of other fluids come into the lumen of alimentary tract. It's always full of water. A lot of our water is actually outside of our bodies. It's inside of the lumen of the alimentary tract. So saliva, stomach juices, pancreatic juices, intestinal juices, bile from the liver. Those are all liquids. They're all mostly water, and they're going to be injected into the GI tract at different points. So there's a large quantity of water inside of the GI tract. Uh, and then, as absorption is happening during the passage through the small intestine and the very beginning of the large intestine, uh, the water is absorbed with it. So the water that was injected into the GI tract gets reabsorbed back into the bloodstream in order that we don't lose gallons and gallons of water every day. And something goes wrong, that happens, right? Your absorption doesn't work very well, and we have diarrhea. We are losing water that way, and we can become dehydrated. Um, the very beginning and the very end of the digestive process are somewhat, at least, under conscious control, thus using skeletal muscles. So, taking the food, biting, chewing, swallowing, are mostly under conscious control. And the very last, defecation, is at least somewhat under the conscious control. The external anal sphincter is a skeletal muscle that we can control. Everything in between, all the movement is controlled by smooth muscles, that thus they are not under conscious control. That's an unconscious movement. Uh, the swallowing, as we talked about in the respiratory system, is a very very complex set of movements by different types of muscles, ensuring that the alimentary tract, the GI tract, and the respiratory uh, pathways that are crossing in the throat don't interfere with each other, especially to ensure that the food does not go into the respiratory tract. So we have the epiglottis closing the top of the larynx. We have vocal cords, both the false and the true ones, tightening, narrowing the passage so the food cannot go all the way into the trachea. We also have the uvula. Uvula is that thing in the back of your mouth that hangs on the back of the mouth. During swallowing, the uvula goes up and blocks the choana. Remember that the opening, the hole between the nasal cavity and the throat is choana. Uvula blocks it. It plugs it, and that way the food that is swallowed doesn't go up into the upper respiratory tract. Inside of the mouth, there are many small salivary glands, like just a cell here and there, and there are three pairs of large glands. Those are the two submandibular glands, two sublingual glands under the tongue, and the largest ones, the two parotid glands. Those are these ones here. They're continuously secreting saliva into the mouth. Saliva contains amylase, which is the very beginning of the breakdown of starch. Starch is a polysaccharide, so when it's broken down into disaccharides and monosaccharides, it's sweet. So when you eat something starchy like bread, the bread itself is not sweet, but as soon as saliva starts working on it, the amylase is breaking down the starch into smaller sugars, and you feel that as sweetness. Saliva also contains some lysozymes and antibodies, which are kind of the first barrier against microorganisms in foreign bodies, so they don't go into the digestive system or into the respiratory system. Parasympathetic system stimulates, sympathetic system inhibits secretion of saliva. So that you get a dry mouth when you're scared. Just think of yourself as a zebra running away from the lion. It will shut down the entire digestive system. All the secretions, all the movement, everything that's happening in GI tract uh, will be inhibited during stress response, thus inhibited by the sympathetic nervous system, thus inhibited by adrenaline and noradrenaline. So chemical digestion of carbohydrates, including starch, starts in the mouth, uh, while proteins and triglycerides start in the stomach. Um, so, for instance, stomach has the enzyme called pepsin, which is the very beginning of breaking down of proteins into smaller peptides, and then those peptides into the amino acids. Uh, everything gets digested in the small intestine using both intestinal enzymes and pancreatic enzymes, mostly pancreatic enzymes. Most of the digestion is done by pancreatic enzymes that are injected into the duodenum, while intestinal cells on the surface of the intestine contain their own enzymes that add some enzymatic activity as well. The bolus from the mouth is pushed down the esophagus by peristalsis. The top third of the esophagus is a mix of skeletal smooth muscles, so you can still kind of force yourself to swallow. Once it gets to the halfway down the esophagus, you don't have any more conscious control over it. You're hoping that gravity and the smooth muscles together, and maybe drinking some water or swallowing more saliva is going to push the bolus in case it gets stuck. Um, and esophagus starts with the upper esophageal sphincter, which is under conscious control. It ends with the cardiac sphincter. That's a smooth muscle ring where esophagus enters the stomach. That's a smooth muscle ring. So it is not under conscious control. This is where the bolus enters the stomach. Stomach is a temporary storage for food. Uh, we eat discrete meals, like we're not grazing around all day long. We tend to have, breakfast, lunch, and dinner, or depends on the culture. Different times of day, we have, sit down and eat a meal. So we have discrete meals, and but we cannot digest and absorb all of those nutrients at once. You have, sit down and have a Thanksgiving meal, which is enormous. This takes some time, uh, and so the meal needs to be contained in the stomach. So our stomach has to be relatively large for our size. In general, carnivores that eat uh, discrete meals and omnivores like us who eat discrete meals have larger stomachs compared to the size of the body than the grazing animals, the herbivores that just graze grass all day long. They can have much smaller stomachs because the food is constantly going through. The stomach is stretchy. It can expand greatly to accommodate a very large meal. Yeah, you feel it. It's not pleasant, but it can. And that stretching is going to result in release of several hormones like gastrin, cholecystokinin, a couple of others that are telling the rest of the body, hey, we just got a big meal, we need to get ready for digestion. Um, Most of the GI tract has the usual layers of muscle, circular layer and longitudinal layer of muscles. Stomach also has oblique muscles, so they go at an angle. So it has another layer. So you have both the circular and longitudinal and oblique muscles in the wall of the stomach, which makes stomach a kind of a strong muscular organ that is churning that food and turning that chyme that is still semi-solid, the bolus that's still semi-solid, into completely liquid chyme. The liquefied chyme is released in small quantities into the duodenum, just a little bit at a time, and through the pyloric sphincter, which is at the end of the stomach. Almost nothing gets absorbed in the stomach. Exceptions are some small quantities of very small molecules that can easily come past membranes. Examples are aspirin and alcohol, but it's still small quantities that get absorbed in the stomach. Most of that is absorbed later. Inside of the stomach lining, there are different kinds of glands. Those glands are, for instance, gastric glands, and they are found in the bottoms of gastric pits. There's a little invaginations inside of the stomach. Uh, these gastric glands produce alkaline mucus, and they produce hydrochloric acid, and they produce pepsinogen. Okay, hydrochloric acid is produced by parietal cells. Uh, Hydrochloric acid activates pepsinogen, turns it into pepsin, and pepsin can start digesting proteins. Uh, Hydrochloric acid is also a defense mechanism. It's one of those barrier defenses. It forms a very acidic environment in the stomach, which kills most of the bacteria, most of the foreign organisms that may enter with food. Hydrochloric acid also denatures proteins, unravels them, which makes them easier for the enzymes to come in and start breaking them down, digesting them. Parietal cells, uh, the same ones that produce hydrochloric acids, also secrete what is called intrinsic factor. Intrinsic factor is the most important chemical digestion function of the stomach. If you remove the stomach, you have to like inject intrinsic factor. Uh, that's You can live without a stomach, but you cannot live with at least some of those parietal cells producing the intrinsic factor. Intrinsic factor is necessary for the absorption of vitamin B12. Vitamin B12, unlike the other vitamins, is an enormous molecule and requires a carrier molecule in order to be uh, kind of latched into the special transporters that are going to absorb vitamin B2 into the cells. That happens at the end of the ileum and beginning of colon. So intrinsic factor is the most important function of the stomach. Secretion of the intrinsic factor, most important function of the stomach, and it is produced by parietal cells, which are the same ones that produce hydrochloric acid. Chief cells are a different type of cells in the gastric pit, and they produce pepsinogen. And pepsinogen is the precursor, the inactive form of pepsin, which is activated by hydrochloric acid and starts breaking down proteins into peptides and peptides into amino acids. There are other cell types here that produce uh, mucus. Some of it is acidic mucus, some of it is alkaline mucus. Um there are cells that produce various hormones like ghrelin, hormones that cells that secrete histamine, serotonin, somatostatin, various different uh, hormones and enzymes that are either needed inside of the stomach or to signal the rest of the GI tract that the meal is here and they better ramp up their function because chyme is coming and needs to be digested. Gastrin acts on the stomach, on the small intestine, and large intestine, and increases the movement, increases the peristalsis. Uh, the acid in the stomach juices is capable of digesting the stomach itself. So the stomach has to protect itself from the acid. So the cells on the surface of the interior of the stomach are protected by the alkaline mucus, which is rich with bicarbonate buffer. So most of the lumen of the stomach is a lot of hydrochloric acid breaking down food, but at the edges, at the where hydrochloric acid meets the cells, there's a thin layer of this alkaline mucus with buffers that is neutralizing the acids right there at the border, right there at the surface of the cell, which means that the HCl is not going to digest that cell, or at least not very fast. There are tight junctions between the cells lining the stomach, which prevents the hydrochloric acid from passing between the cells and digesting the more kind of uh, deeper layers of the wall of the stomach. And stem cells of the stomach are rapidly dividing, replacing the damaged cells. So these cells still do get damaged. I mean, it's digestion is going on, and HCl is there, and pH is somewhere between 1 and 2, and it is going to be very difficult for cells to survive long. So they get replaced all the time. That's the defense from the acid. So when food first comes into the stomach, it sits in the fundus for a while, and salivary amylase is still in that food, and it still continues to break down starches. And then the stomach, moves and mixes and churns that food around and moves that food into the body of the stomach, where it's mixed with the acidic liquid. Uh, the amylase there is inactivated. Uh, but lingual lipase that came out of from the mouth, lingual means the tongue, right, um, is activated by the hydrochloric acid. So, although it was secreted in the mouth, it doesn't start digesting lipids in the mouth. It starts digesting lipids once it's already been in the stomach for a little while. And that lipase, once activated, is starting to digest triglycerides. Um, so there's pepsin from the stomach, uh, lingual lipase from the mouth, digesting triglycerides, and the salivary amylase for a little while still breaks down starches, but then it gets inactivated. So that's the extent of kind of chemical breakdown of foods in the stomach. Actually, not that much. So liquefied food, the chyme, has gradually moved toward the pyloric sphincter, the pylorus, and the pyloric sphincter allows only about 2 to 3 milliliters of chyme to exit the stomach and enter the duodenum. This is called gastric emptying, and it doesn't happen very very fast. Let a couple of milliliters go, and then stop and wait, and then after a couple of minutes, maybe another 2 or 3 milliliters. So it's really releasing only small quantities of that highly acidic juice into duodenum. Duodenum is unprepared for that kind of uh, that kind of acidic liquid, right? That's like it's gonna burn itself if you have more than a couple of milliliters. But you can uh, let 2 or 3 milliliters because at the very beginning of duodenum there are three sources of buffers. There are buffers containing bile that's coming from the gallbladder and the liver. There's buffer coming from the pancreas together with the pancreatic juices and other enzymes. And bicarbonate buffer is also secreted by the Brunner's glands that are in the lining and the epithelium of the duodenum itself. So as soon as that acidic liquid in a very, very small quantity, 2 or 3 milliliters, comes into duodenum, the first thing the duodenum does is neutralize it. And after that comes even a little bit alkaline, a little bit higher than pH of 7. Um, so the meal stays in the stomach for quite a while. Depends on how big the meal is. A huge meal is going to stay longer than a small meal. Also, composition of the meal. If you just eat a bar of chocolate, it's just a bunch of carbohydrate. It doesn't stay very long. It gets liquefied and moved on pretty quickly. Protein-rich meal stays in the stomach a little longer. The fatty meal stays the longest. And in general, digestion of fatty meals, the meals that contain a lot of lipids, is going to last the longest, not just in the stomach, but in the entire GI tract. Uh, so a fatty meal is going to stay in the stomach up to 6 hours. That's quite a long time. The total duration of digestion from biting the food to pooping is anywhere between 24 and 72 hours. So even if you have a very, very fatty meal, 6 hours in the stomach is only a small proportion of the time that the food is going through the GI tract. And if it's a high fat meal, it's going to stay longer in the stomach, but it's going to stay even longer in the rest of the GI tract. Okay, most of the digestion, chemical digestion and physical mechanical digestion, and almost all of absorption happens in the small intestine. It is small not because it's short, it's actually very long. It's about 10 feet long, 3 meters, but because it has a small diameter of the tube. It's a narrow tube. That's why it's called small. It's a thin intestine, really, rather than small. The main parts of the small intestine are the duodenum, jejunum, and the ileum. Duodenum is about 12 inches long. That's actually what the word duodenum means, 12 inches. Jejunum is about 3 feet, and ileum is about 6 feet. And small intestine has all sorts of folds and villi and microvilli on the surfaces of the cells. And so the surface area is greatly increased by all of those folds and villi and microvilli. So the surface area of the inside of the small intestine is around 200 square meters. That's quite a lot. That's what, half a soccer stadium? Um, so this surface area allows for all the digestion and absorption process to occur efficiently. It's just like in the respiration, it's the surface area of the lung, of the alveoli, that allows all the gases to pass, and not so much the volume. The same thing is with intestines. It's the surface area of the inside of the intestine that allows all those molecules to pass between the food and the cardiovascular system. Uh, let's go back to the duodenum and the very beginning of it, because the very beginning of the duodenum is one of the most intricate and most interesting parts of the GI tract. Um, the very first part of the duodenum, let's say first inch out of the 12 inches, is what is called ampulla of Vater. Ampulla of Vater, or hepatopancreatic ampulla, is the place where you have Brunner's glands that secrete buffers that neutralize the acid. This is where the pancreatic duct comes in from the pancreas, and brings in all the pancreatic digestive enzymes as well as buffers. And this is where the bile duct comes in, bringing in the bile from the gallbladder and from the liver. So pancreatic duct and bile duct are emptying into the duodenum right next to each other, right under those two openings of the pancreatic duct and bile duct is a sphincter of Oddi. Sphincter of Oddi controls the movement of these juices through the ampulla of Vater into the lower parts of the duodenum. It's essentially keeping that 2 to 3 milliliters of chyme that just came in from the stomach through the pyloric sphincter. It keeps it in the ampulla of Vater for a little while until it's completely alkalinized, that acid is neutralized, and now pH is higher than 7, before it allows it, the sphincter of Oddi opens and allows it to go into the rest of the GI tract. Um, so, pancreatic enzymes are, as I said, essential for the subsequent digestion. Most of the food is digested by the enzymes coming from the pancreas. Much smaller proportion of the molecules are digested by the enzymes that are secreted by the enterocytes themselves, the cells on the surface of the inside of the intestine. The bile coming from the liver and from the gallbladder is necessary for digestion of lipids. So ampulovator is important because all of these processes are happening there. So the complete digestion of everything can now start because pancreas, gallbladder, liver, and Brunner's glands, stomach, everything has now put in their stuff in there, and now we can start digestion and absorption in the small intestine. Um, the small intestine, as I said, has folds, circular folds that have villi, and one villus has a lot of cells on its surface, and each one of those cells has the microvilli villi, and then they have little villi on the apical surface of the cell. Uh, Between the two uh, villi are what are called the crypts of Lieberkühn. The crypts of Lieberkühn are also kind of little canals lined by various kinds of cells. Those different types of cells are the cells that secrete digestive enzymes, secrete some hormones, and some of them are parts of the immune system protecting the intestine from microorganisms. Okay, so carbohydrates, proteins, and nucleic acids are digested by pancreatic enzymes and enteric enzymes, uh, and it's relatively simple. You break down large carbohydrates, polysaccharides into disaccharides, and those into monosaccharides, and then monosaccharides, like glucose, get transported, actively transport over the surfaces of the cells into the cells, and from there into capillaries. The same thing for nucleic acids. Nucleic acids get broken down into A's, T's, C's, and G's adenine, guanine, and cytosine, completely separated from each other, separated from the phosphate, separated from the sugar, the backbone sugar, the ribose or deoxyribose. So everything is broken down into tiny little pieces before it's absorbed into the intestinal cells, and from there into the capillaries. Uh, Proteins are broken first, from large proteins into smaller and smaller peptides, and then peptides are broken down into the 20 essential amino acids. Every amino acid is individually absorbed by the intestinal cells and put into capillaries. All those things are now in the capillaries. They are the capillaries of the hepatic portal system. Hepatic portal system is going to bring all those little, breakdown products of digestion through the bloodstream into the liver. The hepatic portal vein is going to enter the liver, bringing all that blood and all that extra liquid, remember, that was in the GI tract, plus all those little breakdown products of food, the little amino acids and the little monosaccharides and the little purines and pyrimidines and phosphates and sugars, and All those things are going to end up in the liver, where the liver is going to process it. If any kind of molecule managed to pass through because it's toxic and we don't really have kind of a mechanism to block it or to digest it or anything, it just kind of enters the cell and gets into the bloodstream. It is going to be detoxified by liver. All sorts of you know things are going to happen in the liver, like these are now Lego blocks. Let's build human proteins and human DNA and human RNA and human sugars, and so liver is kind of very busy getting all those digestive products from the intestine via the hepatic portal circulation, via the hepatic portal vein. uh, Remember, liver also gets blood from the hepatic artery. Hepatic artery is tiny. Hepatic artery is a small little branch of what, the celiac artery, and celiac artery is a small thin branch of the abdominal aorta. And so liver is getting much more blood from the portal system than from the hepatic artery, which means it's actually getting much more oxygen from the venous blood than from the arterial blood. Remember the whole oxygen dissociation curve from the last unit and the respiration. Well, most of the red blood cells and most of the hemoglobin, even when they're deoxygenated, still have a lot of oxygen in it. So oxygenation and deoxygenation of hemoglobin is like really removing some oxygen here and there, but mostly they're saturated. So the venous blood coming from the GI tract is also going to be mostly oxygenated, mostly saturated, not fully saturated, like an arterial blood would be, but saturated enough that it can have enough oxygen to give to the liver for all of its busy work. Hepatocytes are very metabolically active cells, and they are building the liver and doing all of that work, metabolic work, anabolism, catabolism, detoxification. Okay, so what about the fats? The lipids, well, you have triglycerides and you have steroids. There are different types of lipids. They are complicated, they are large, and they are difficult to digest. First of all, lipids are not solvable in water. You have lava lamps, right? How do you make a lava lamp? You mix some water and some oil. And so oil and water don't mix, which means when you eat a fatty meal, all those lipids are going to glom together. They're going to make a big globule. And poor lipase is coming from the pancreas or the lingual lipase or from the enterocytes themselves. They can attach themselves from the outside and barely begin any kind of digestion of that big lipid globule. There's too little surface and too much volume, and so we want to reduce. We want to increase the surface to volume ratio. We want to have much more surface where lipases can attach and much less volume, so they don't have to dig deep. There's not going to be lipids that are going to hide in the middle and be unreachable. And how do we do that? Well, how do we do that in our households? We use soaps and detergents to break down fatty stuff. And our bodies produce salts and detergents as breakdowns of many different biochemical reactions happening in the liver. And so those kind of breakdown products that act as soaps and detergents, those are, one word for them are bile salts. Bile salts are going to be secreted by hepatocytes, put in a tiny little bile canaliculi that are going to join into the left and right uh, uh, hepatic duct. Those two ducts, one in the left and one in the right lobe of the liver, they're going to fuse into the common hepatic duct. Some of those bile canaliculi, instead of going into the hepatic duct, into the common hepatic duct, are actually going to go into the gallbladder. They're going to start filling up the gallbladder with bile. And gallbladder is just a sac, right? A cyst. It's a little balloon, which as it's filling with with bile is going to expand, get bigger and bigger because it's now full of liquid. Coming out of the gallbladder is a cystic duct. Six cystic duct and common hepatic duct fuse into what is called a common bile duct. Common bile duct goes where? Goes into the duodenum, into the ampulla of vata, right next to the pancreatic duct. So that's the way that the bile gets into the duodenum, gets into the GI tract. All those soaps and detergents are dripping from the common hepatic into the common bile duct, into the duodenum all the time. Drip, 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 drip. And if there are any lipids in the intestine, those salts are going to break down the big globule of fat into lots of little droplets of fat. Now there's a lot more surface, a lot less volume, and so the lipases can attach those molecules and start breaking them down and digesting fats. If you eat a big fatty meal, uh, the stomach, especially the end of the stomach, the pyloric region, is going to secrete a hormone called cholecystokinin. Cholecystokinin is a hormone that is going to stimulate the gallbladder, the smooth muscles in the wall of the gallbladder, to squeeze, to contract, and to squeeze all that bile out of the gallbladder through the cystic duct into the common bile duct into the duodenum. So instead of just drip, 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 you have suddenly a large quantity of bile going in at the same time when a large quantity of semi-digested fatty food is coming out of the stomach into the duodenum. So all those big globules of your lava lipids out of the duodenum get broken down in tiny little droplets, which now your lipases, intestinal lipase, lingual lipase, and pancreatic lipase can now attach to and break down. Uh, Animals that don't eat big meals, big discrete meals, fatty meals, lots of meat, lots of fat, like us, they don't need the gallbladder. Horses eat grass all day long. They eat hay all day long. There's not much fat in there. And they do all, they eat essentially 24 hours a day. And the little drip, drip, drip from the common hepatic duct is all they need. And so they don't have a gallbladder, they don't have a cystic duct. They just have a common hepatic duct, which becomes on its own the common bile duct, or just bile duct, and drains a little bit of bile all the time, drip, 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 into the duodenum. We eat discrete meals, we eat a lot of meat, we eat a lot of fatty stuff, and thus, we need a gallbladder because we need a lot of those soaps and detergents at the same time when we swallow a big fatty meal. All right, even when the lipases are breaking down those, phospholipids and various kinds of lipids, that's very complicated. They do get transported into the enterocytes, and the enterocytes reconstitute triglycerides, right? The glycerol and the three fatty acids reconstitute them inside of the cells. So those are now big fatty molecules inside of the enterocytes. It's going to be very, very difficult to put those things into the capillaries. Capillaries are too small, these molecules are too big. And so instead of the capillaries of the hepatic portal system, the fats that were just a result of the digestion go into lacteals. Remember, enterocytes have built our versions of fats. It doesn't matter if the fat came from a cow or artificial fat, margarine, doesn't matter. It's going to break it down and then build inside of the cells of the intestine. It's going to build our versions of fats. But those versions of fats, those molecules are too big to go into the capillaries, so they go into the lymphatic system. Lacteals are lymphatic capillaries. And the lacteals are going to go where? The rest of the lymphatic system. They're going to go through a lymph node for, kind of cleaning and making sure there are no microorganisms there, and then they end up in the left and right subclavian veins, which means they are going to go into the circulation in a big uh, blood vessel, not a tiny capillary. So the the lipids don't go through the liver first. They go straight into the, venous circulation, from there into the heart, from the heart into the rest of the circulation. A lot of it is going to go on first, second, third pass. It's going to go through the liver, where the liver can deal with those lipids then. So uh, most of the stuff goes to the liver directly. Lipids don't go to the liver directly; they go indirectly after they pass through the systemic circulation one, two, three, four times. All right, the large intestine: cecum, appendix, colon, rectum, and anus. There is an ileocecal valve, which is a valve that controls the movement of chyme from the small to large intestine. In humans, cecum is small, mostly absorbs water and salts. Um, in other animals, cecum can be very large. In horses, in animals like horses, donkeys, tapirs, rhinos, cecum is a meter long. It's an enormous structure. It's a fermentation vat where that's where all the bacteria and protists and fungi and all the other microflora live and help with the digestion and detoxification and breakdown of cellulose uh, and production of vitamins that is happening in the cecum of that type of animal. In us, cecum is small, doesn't do much. Most of our cecum is actually shrunk and shriveled, and forms what is called the appendix. Most animals don't have appendix. We do have an appendix. An appendix has lymphatic tissue, and it may also serve as a reservoir of intestinal flora. Our intestinal flora, our fermentation vat is the colon, so it comes after. And so, in case a disease wipes out your intestinal flora, or a course of antibiotics wipes out your intestinal flora, there's going to be a source. Some of those bacteria and protists are going to be sitting in the appendix, and once it's all clears out, they're going to reproduce and start recolonizing your colon, where most of your uh, microflora lives. Um, Colon is going to finish absorbing the water. About 90% of the water has been already reabsorbed by the small intestine. The remaining 10% is reabsorbed by the colon. If something doesn't work that well here, you're going to have diarrhea. Uh, there are some bacteria there, and some of those bacteria are producing vitamins, and those vitamins get absorbed in the colon. And as the stuff goes through the colon, it forms feces, and from there it goes to the rectum and through the anus outside into the outside world. Anus has two sphincters. Internal sphincter is smooth muscles, not under conscious control. The external sphincter is skeletal muscle that is under conscious control. So that's how feces goes out. Um, the colon has strips of fibrous connective tissue, tania and haustre. Tania are longitudinal. So if this is the colon, you'll have a tania here, and a tania here, and a tania here, and a tania here. Haustre are circular. They're going like this, and like this, and like this. So as the food, the food that is gradually turning from the liquid chyme into the solid feces, as it's being moved, pushed back and forth by peristalsis of the colon, the tania and caustre, by uh, contracting and uncontracting, are going to form the shape of the feces. Now, human feces doesn't have much of a shape. It has the shape of a rectum. But a lot of other animals, if you go out in the woods, you're going to recognize, hey, this is poop from a deer, and this is from a cat, and this is from a dog, and this is from a horse, and this is from a cow, and this is from a rabbit. You're going to recognize different types of feces from different other animals. And it's the tania and haustre, how they're organized and how they're controlled, that determines what is going to be the shape and size of the pieces of feces in every different species. And this has been studied most recently in an Australian animal called a wombat, because the wombat poops square cubes. It poops. A lot of animals leave poop in particular strategic places as marking their territory. Wombats need to mark the territories on the tops of rocks. And if you poop something that is a sphere, that poop will roll off the rock. If you poop out the square, if you poop a cube, you are going to have that cube sitting on the top of the rock, telling all the wombats, "Hey, this is my rock." And so scientists over the last few years have been studying exactly how the colon and the rectum, the taenia and haustra of the colon and the rectum, are shaping the feces, and learned a lot about the processes that go on that. It has nothing to do with the anus itself. It has to do with the taenia and haustra. Okay, so um, you need to know the basic anatomy and histology of the liver, the hepatocytes, the central vein, and the bile canaliculi. How it's all organized. Also in the lab, be able to recognize liver, those hexagons and pentagons of the liver lobules under the microscope. Uh, and everything about the gallbladder, the common hepatic duct, the cystic duct, and how they fuse into the common bile duct. Those are kind of a lab things, as well as the lecture questions. Okay, pancreas. Pancreas is the main source of digestive enzymes, as I said, and and enzymes that digest carbohydrates, lipids, starches, nucleic acids, so various, amylases, nucleases, lipases, uh, uh, are going to be made in the pancreas in their final form, the active form, and just secreted into the duodenum. No big deal. On the other hand, the enzymes that break down proteins, like trypsin, uh, they are not made in an active form. They are made in an inactive form because an active form of trypsin would digest the pancreas from within. You don't want that to happen. So trypsinogen, an inactive form of trypsin, gets secreted into the intestine. And there it is activated and becomes trypsin. And activated trypsin not just digests the uh, the uh, the proteins, but also leads to the activation of other enzymes that break down proteins, like chymotrypsin and carboxypeptidase. There are several different enzymes that break different kind of parts of proteins or types of proteins and cleave different amino acids from each other. So anything they break down, proteins has to be secreted by the pancreas in an inactive form and activated in the intestine. All the other types of enzymes are ready-made active versions that are coming out of the pancreas. Um, okay. Vitamins are mentioned. They're mostly absorbed at the end of the ileum or in the colon. Doesn't matter, some of them are coming from the food, some of them are made by our microflora that lives in our colon. Microflora, as I said, in us lives in the colon, and things like horses and rhinos in the cecum. Remember that animals that chew cud, various, cows, sheep, goats, camels, giraffes, antelopes, all sorts of things like that. They have a very complex stomach. They have four chambers of the stomach. Only the fourth chamber is equivalent to our stomach. And the first three are chambers for different types of fermentation. And the first one contains a lot of various microorganisms. The microflora that really does the job for cows is in the very beginning. It's the first chamber of the stomach, which is why cows can detoxify. Those microorganisms can detoxify all sorts of stuff for them. They can eat really moldy hay and be okay. You give moldy hay to a horse, and the horse is dead because the toxins are going through the small stomach and the long small intestine and the large intestine, finally gets to the cecum where it can be detoxified. By then that time it's too late. The same thing with us. We can't eat toxins because our microorganisms that in cows protect them right at the beginning of digestion are in the colon after all the digestion and absorption has essentially been finished. Uh, as I mentioned before, uh, the intrinsic factor from the stomach is a transporter molecule that binds to the vitamin B, which is a very large molecule, and only this complex, the intrinsic factor plus vitamin B complex, can be transported into the cells by endocytosis, and that happens at the end of the ileum, and that's how it goes into the circulation straight to the liver. About 9 liters of water enter the intestine every day, and 90% of them gets reabsorbed by the small intestine, about 10% by the large intestine. Very little goes out with the feces. As far as metabolism, there's a large chapter on metabolism. All I want you to know is the difference between anabolism and catabolism, and a couple of hormones that stimulate either anabolism or hormones that stimulate catabolism. Gastrointestinal tract produces large quantities of serotonin, which acts as a local neurotransmitter. Remember that there's a third brain in the intestine. That the intestine has its own kind of semi-independent nervous system. The intestinal nervous system uses serotonin quite a lot as its neurotransmitter. Serotonin stimulates peristalsis, and serotonin stimulates secretions. Now remember, one of the main breakdown products of serotonin after just two enzymatic reactions is melatonin, which means a lot of melatonin is going to be produced in the intestine. It is mostly not going to get into circulation. It's going to act locally. How is it going to act locally? During the night at large doses, melatonin is going to have a stimulating effect on digestion, especially on the peristalsis. At small doses, when you have really low doses of melatonin, which is during the day, it has inhibitory effect on on peristalsis. This way, the melatonin secretion in the intestine itself is synchronizing kind of the circadian rhythm of digestion. We digest actually more and more actively during the night while we sleep than during the day when we are active. Speaking of circadian clocks, they control daily rhythms of everything in our bodies. So they control the rhythm of liver. Liver is doing what? It's building stuff, breaking down stuff, detoxifying toxins. How does it do that? By producing enzymes that can do all those things, right? Where do the enzymes come from? Well, they're proteins, which means they are the result of transcription of genes and translation of RNA into protein. So what the circadian clock in the liver does, it turns on and off those genes, which means some of those enzymes that are doing particular biochemical functions are active at different times of the day, others at other times of the day. So, for instance, the example I give you in my study guide, and there's like, I don't know, 10, 15 links that I sent in the last email, and that you can find also in additional readings that are talking about different aspects of this. But let me kind of simplify it, that the circadian clock in the liver is turning on the gene that codes for the enzyme called alcohol dehydrogenase in the early afternoon and shuts it down about midnight. So there's about a 12-hour period from about noon to midnight when alcohol dehydrogenase is produced by the liver. From midnight to noon, it's essentially shut down. There's no alcohol dehydrogenase. Now remember, a little bit of alcohol, a tiny little bit of alcohol, is absorbed by the stomach. That's not enough to go all straight to the brain and make you drunk. If you have an alcoholic drink in the afternoon or the evening, which is when most people do, uh, that alcohol is going to be absorbed from the intestine, go into the hepatic portal system, goes into the liver, where it's going to encounter alcohol dehydrogenase, which is wonderfully active at that time of day, and is going to be broken down. You need to drink enormous quantities before you overpower the liver. There's not enough alcohol dehydrogenase to detoxify all the alcohol that you drank. That was not very responsible drinking. And so finally, some of it leaks into the circulation, passes the blood brain barrier, and you feel the effects. People who are addicted to alcohol, the true alcoholics, they drink in the morning. They need that hit. They wake up first in the morning and they have a craving for alcohol, right? They need a dopamine hit. They need alcohol to hit the brains as soon as possible, and so they're drinking in the morning, because in the morning that gets absorbed through the intestine, and they don't eat much in the morning, so it's all empty, so the alcohol goes pretty quickly through the stomach, through the intestine, into the circulation, into the liver, through the liver, into the main circulation, through the blood barrier to the brain, so they can get a hit fast. Which is why social drinking is reserved for the evening, because we can have a beer or two and still be okay. If you have a beer or two in the morning, you're going to definitely feel a strong buzz, because there was no alcohol dehydrogenase transcribed and translated in the liver in the morning. And this is already very long, so I'm going to stop here and see you in the next video.